Hello, it's Danyang speaking here. I'm a PhD student in University of Copenhagen. I will share my ongoing PhD project, Structural and Functional Characterization of the Apical Domains of Human CCT, which under the supervision of Professor Guillermo Montoya. We are all familiar with the central dogma of molecular biology. The main information flow is from DNA to RNA and then to protein. But we miss a really important information here is we don't really know how the nascent peptide falls properly into the functional state. We all know the structure determines the function, so it is really important when the protein falls properly into its native state to form the function in the cell. But some of the protein can form themselves, which is spontaneously processed by hydrophobic interactions, formation of hydrogen bonds or van der Waals forces, and so on. But there are 5 to 10% newly synthesized polypeptides in eukarya need help, which means they cannot form spontaneously by themselves. And CCT is a group 2 chaperoning, which is required for the folding of large and growing number of proteins. It has been estimated that approximately 10% cytosolic proteins interact with CCT, and some of these substrates involved in vital cellular function, such as cytoskeleton formation, protein degradation, and cell cycle control. That's a reason as a key regulatory hub, the mechanism of CCT is really important to reveal not only the cystate folding process, but also the hint how to interfere and inhibit the folding process of specific substrate. CCT is a 1 megadalton complex which composed of 2 copies of 8 different subunits. And 8 different subunits form a superposed ring, which you can see in the figure, and 2 superposed rings stacked back to back, forming 2 cavities where the folding process takes place. And CCT switches the open substrate receptive conformation to closed substrate capture conformation powered by ATP hydrolysis. Although the subunits are quite different and divergent, but they share really similar structures. And each subunit has apical, intermediate, and equatorial domains, which you can see in the right side figure. And the most conserved region is the equatorial domain, which ATP binds to power the whole folding process. And the most flexible and divergent part is the apical domain, which is facing to intracellular environment and is believed to the part to recognize and bind different substrates. And how does CCT recognize a substrate? The previous work showed that the apical and the sensor loops involved in the binding of tubulin. It's a famous substrate. This is the cryo map of East CCT, which you can see in the central part, and the conserved equatorial domain fits quite well with the density, but at the apical parts is quite they cannot quite fit quite well due to the apical domain. They are quite divergent and flexible, which also fits the function of apicals, which can recognize lots of different substrates and quick capture and quick release them. Therefore, my objective is to express and purify 8 different apical domains as well as the selected substrates, and then by structural and functional analysis to understand how exactly apical domains can recognize and bind with different substrates. Take apical 3 as an example. After expressed in E. coli system, the protein was purified by affinity and size exclusive chromatography, and which you can see it has a nice peak showed by gel filtration and corresponding to the SDS page single band. The quality of the protein is controlled by mass spectrum and circular dichroism to confirm the primary and secondary structure of the protein. After get the relative pure protein, the crystallization experiment was set up. And luckily, I get the plate-shaped crystal of Apical 3, which diffract to 3 angstrom with a native protein. At the same time, the selenomat crystal diffract to 3.8. By employing the molecular replacement and also the experimental phase and set, I got around 3.4 structure here. In addition, I also got crystals for Apical 4, Apical 6, and Apical 8, both with a native protein and also selenomat protein. The data processing is ongoing with this part. And another part of my project is the interaction between apical domains and substrates. From the literature, VHL is a tumor suppressor protein interact with CCT1, and another protein is P6, which is a Dawkins side of HIV1 and interacts with Apical 3. That's the reason I take VHL and P6 as a starting point to investigate how apical and the substrate bind together. After confirmed by a pull-down assay, after the interaction confirmed by the pull-down assay, I try to co-crystallize the apical domains and substrates. Here are the hits I have obtained and try to optimize. So the crystal of Apical 3 plus P6, which diffract to 3.6 angstrom, which is not really high resolution, and unfortunately from the map here, I can only say Apical 3 but not Apical 6. The same story happened with Apical 3 and VHL, which is the crystal diffract to 3 angstrom, but unfortunately, I cannot see VHL in the density map. So for future plan, I will try to finalize the structure of apical 4, 6, and 8, and try to fit the apical structure into the cryo-EM model of the whole CCT to get a better view of how CCT structure is and how exactly the mechanism behind. The other side, I will try to optimize the co-crystallization of apical domains and substrates, try to figure out what exactly the binding mode. And in the end, I would thank my supervisor and also all the people in the lab and the facility, especially our 2 rock imagers in the lab. Without the help of the rock imager, I cannot screen more than 100 plates by myself. Thank you for your attention. That's my talk.